Cells you should have talked about in Bio 111, so I don't want to talk a whole lot about them, but we just need to be aware that cells are the basic living unit of life. So cells are going to make up our tissues, tissues make up our organs, and that cells are the smallest living organisms that display all of the characteristics that are required to be considered alive. So cells are the microscopic basis for living organisms. We have over 200 types of cells in the body. So we'll talk about cells more when we talk about tissues. But as far as anatomy and physiology goes, we have different shapes of cells. And the different shapes of cells and the different organelles of cells are going to give different types of cells their qualities that help them give tissues their particular characteristics. So we've got some cells that have weird shapes, they're kind of elongated, have extensions like fibroblasts we find in connective tissues. We've got biconcave disc cells, these are erythrocytes, these are your red blood cells that carry oxygen. We've got cells called epithelial cells. Epithelial tissues line and cover all of your body surfaces. So we've got different types of cells that make up our epithelial. We'll talk about those on Monday when we start talking about tissues. We have cells that help with movement. Our muscle cells, we've got skeletal muscle cells, which are actually big, long fibers. The cells are as long as the muscle that they live in. You have cardiac muscle cells that are continually pumping to move blood through your heart. You've got smooth muscle cells that you find in the walls of your hollow organs to move things through your organs. You've got cells that store nutrients. Fat cells are called adipocytes. They accumulate large fat droplets that can be accessed in time of nutrient deprivation. Also, bone cells store high levels of minerals, so that would be another example of a cell that stores nutrients. We've got cells that fight diseases. This is an example of a cell called a macrophage. Macrophages are big eaters. They gobble up things that are foreign. We've got cells that gather information and send signals, neurons. Nerve cells are really exciting. We'll talk about those. And then we've got cells of reproduction. These are haploid cells. They have half the chromosome content. And we've got sperm in males and ova in females that are able to pass on our genetic information. So cells are pretty diverse. In general, we can look at a general schemata of a cell and say in general cells have organelles that are going to be specialized from one cell to another. They all have a plasma membrane that's a phospholipid bilayer that keeps them separate from the rest of their environment. If we look inside the cell, the plasma membrane is really important because it's forming the barrier between the internal and external environment. In the plasma membrane, we can have proteins that act as like channels, they can act as receptors for signaling. You can find lipids in the phospholipid bilayer. Most of them are phospholipids, but you can also find cholesterol that helps to give it this fluidity. And really the importance of this plasma membrane is keeping this barrier between internal and external. So we're going to look at differences between like extracellular fluid concentrations of things and intracellular fluid concentrations of things, but we'll go through that specifically when we get to specific cell types. So again, your plasma membrane has lipids in it. They're going to help with that fluidity. It's got proteins that help with signaling or identification. We don't have enough time to go through all the functions. You can kind of just review it. We do need to talk about cell junctions a little bit because this is going to be how our cells link up together to form a tissue. The first type of cell junction that we have is what's called a tight junction. And in a tight junction, this is where you're really snapping yourself, like sewing yourself together so that nothing can get through. So tight junctions are impermeable. And you need to have tight junctions in places where we don't want things to be able to passively flow between cells. So the blood-brain barrier is composed of tight junctions. The blood-testes barrier is composed of tight junctions. In the stomach, your epithelial cells are joined by tight junctions. So that you're not absorbing things through the stomach, you wait until you get to the small intestine to start absorbing things. So tight junctions are really going to hold cells tightly together. The next type of junction we have is what's called a desmosome. And desmosomes they snap cells together. And so they're holding them together, but not as tightly as a tight junction. They're not fully impermeable. They anchor our cells together so that like your skin can move as a cohesive tissue because the epithelial cells in your skin are held together by desmosomes. So they just kind of snap things together, but not so tightly as tight junctions do. The last type of junction that's going to be important is what's called a gap junction. And these are chemical junctions. They're like holes that happen between cells. So you can see here we've got these pores between cells, and what that allows for is instantaneous communication of a chemical from one cell to another. So these are pores between cells. And like if we look at our heart, the heart has a combination of desmosomes and tight junctions, a specialized structure we call an intercalated disc. And what that's going to do is hold all of the cells in your heart together physically and chemically, so that when one contracts, they all contract. And if that doesn't happen efficiently, you have inefficient pumping of blood. Okay, you probably went over membrane transport in Bio 111, so you can kind of review those as well. We've got passive and active processes. Passive processes do not require ATP. They're working based on a chemical concentration gradient. So simple diffusion is going to be when you have molecules like lipid-soluble molecules can slide right through your phospholipid bilayer, and they'll do so down a concentration gradient. We can have carrier-mediated diffusion where you've got a protein stuck up in the membrane, and it's also going to move things down their concentration gradient. So passive processes are always moving down a concentration gradient. We can also just stick a channel in the membrane and let things flow through it. So like water, it has it's an amphipathic molecule that's got different parts that feel different charges. So it's not easy to slip through, but if you stick a protein in there called an aquaporin, then water can go down its concentration gradient. Osmosis, so this would be an example where we've got this channel-mediated diffusion of water. The diffusion of water is specifically called osmosis, and water always moves from what we call a hypertonic solution to a hypotonic solution. So it's going to also go down its concentration gradient. Why is this important? It's important for body cells. I don't know, if we stick a red blood cell in an isotonic solution, then you'll have a net movement of water in and out. If you stick it in a hypertonic solution, where there are more solutes outside the cell than in, water is going to leave the cell and it will shrivel. If you stick it in a hypotonic solution, then water is going to move from it, where you have more water out here, it'll move into the cell, it'll swell. We can just review a little bit about, I'll actually pick up here on Monday, because we do want to talk a little bit about our active processes, because they're going to be important when we talk about like nerve signaling. And you are going to have to understand those a little bit in a little bit greater detail for nerve signaling. So we'll pick up here on Monday, and then we'll continue talking about tissues.